infection in the placenta can cause inflammation, and this helps to fight off infection, but it can also have detrimental effects on the pregnancy, such as preterm labor. One of the functions of the placenta is to detect these infections, and it does so through its many receptors, including knot-like receptors, or NLRs. These detect infectious components in the cytoplasm. IL-1 beta is one of the cytokines that can be produced when an infection is detected. It's very closely regulated because it causes an extremely intense inflammatory response, and it is known that this extreme inflammation can cause preterm labor in vivo in mice. I mentioned NLRs earlier, one of which is NAL3, which works through the NAL3 inflammasome, which you can see here. This is thought to work together to activate IL-1 beta. I'll now go into detail about each step of this process. The first step is when NAL3 detects an infectious component in the cytoplasm. This triggers ASC, which then in turn activates caspase 1. Caspase 1 then transforms pro-IL-1 beta into active IL-1 beta, which can then be secreted through the membrane, causing an inflammatory response. Previous experiments in the Abraham's lab have shown that NAL3 and ASC are present in placenta or in trophoblast cells, as you can see here by these Western plots. Another experiment in the Abraham's lab has shown that MSU, while not a bacterial infection, has the same effect, and it causes an increase in the concentration of active IL-1 beta, as you can see here. So my hypothesis was that when an infectious component is introduced to trophoblast cells, the active IL-1 beta that we know is being produced is being created through the inflammasome. My objective was to show that when the ASC gene is silenced by shRNA, there is not the same increase in active IL-1 beta when MSU is introduced to the cells, and this is because the inflammasome is unable to function without ASC. I mentioned shRNA. First, I have to define wild type. Wild type is the normal non-mutated form of a gene, in this case of ASC. shRNA, on the other hand, switches off or silences a gene. In other words, there will not be any ASC in the cells, and the inflammasome cannot function. So for every trial of my experiments, I used 4 different types of SW71 trophoblast cells. The first was a wild-type cell line with no treatment, then wild-type treated with MSU, and then 2 types of shRNA cell lines, one with MSU and one with no treatment. The first step was to collect the samples, which I did through cell lysis and supernatant collection. Cell lysis collects the proteins that are inside the cell, while supernatants collect the proteins that have been secreted. The next step was to do a VCA, which is done on the lysates and measures the total concentration of protein in the sample. Next are the ELISAs, which is similar to a VCA, but is done on the supernatants and measures the concentration of a specific protein in a sample. Finally, there's Western blots, which compare the relative amount of a specific protein to other samples. And this is also very convenient when there is more than one form of a protein, say a pro and an active form. So my first step was to prove that shRNA was working. You can see in the top line, this is beta-actin, and this is here to show that there is sample in all of the wells and that there is protein in all of the samples. But if you look at the ASC line, there is protein in the wild-type cells, but not in the shRNA cells. So this is proving that the ASC gene has been knocked down. Then I went on to show that the inflammasome is indeed not functioning when tested for IL-1 beta, and you can see with the wild type cells, there is clearly an increase, a significant increase in IL-1 beta when MSU is added. And while there's also a significant increase in IL-1 beta when MSU is added to the shRNA cells, by comparing the two types of MSU cells, you can see that the increase is not nearly as profound or significant. So therefore, IL-1 beta production is being inhibited by the shRNA. Finally, I did a Western blot for IL-1 beta, and you can see with the wild type, there is more pro-IL-1 beta for the no treatment than the MSU, while the reverse is true for the active. This is because when MSU is introduced to the cells, more pro is being activated so that it can fight the infection. This is not the case, however, for the shRNA cells, where there is not an increase in active IL-1 beta for the MSU cells, and there appears to be a decrease. So, in conclusion, the hypothesis was correct in stating that the IL-1 beta is being activated by the inflammasome, which works when an infection is detected by NAL3, triggering ASC, activating caspase 1, which then activates IL-1 beta, so it can be secreted and cause an inflammatory response. My experiments have shown that when ASC is not present, and the pro-caspase 1 cannot become active caspase 1, there will not be an inflammatory response